Good morning, dear students. Today we will study the mechanism of interaction of allelic genes as the basis for the formation of normal and pathological signs of a person. Inheritance is a way of transmitting hereditary information that changes depending on the forms of reproduction. In asexual reproduction, inheritance occurs through vegetative cells and spores, providing great similarity between maternal and daughter generations. During sexual reproduction through the sex cells, the similarities between parents and children are less, but the variability is great and there is a wealth of material for selection. Inheritance is a way of dis- distributing hereditary information from parents to children, depending on the location of the hereditary material in the cell. Nuclear genes are located in the chromosomes in the nucleus, and cytoplasmic genes are in the DNA or organelles. Inheritance are distinguished. In higher eukaryotes, gametes are formed and the cytoplasm is transmitted by female reproductive cells. In these organisms, cytoplasmic inheritance is characterized by a maternal effect. Only the characteristics of the mother and transmitted through the cytoplasm. The set of hereditary inclinations of the cytoplasm is called plasmon, and the inclinations themselves are called plasma genes. Mitochondrial genes can mutate and cause some malformation in humans. For example, fusion of the lower limbs, bifurcation of the spinal column. There are several types of traits inheritance. Nuclear inheritance can be autosomal. Genes are found in autosomes, and sex-linked. Genes are found in the sex chromosomes. Information about the mechanisms of inheritance is important in medical genetic counseling when determining the risk of having a child with a hereditary disease. The qualitative characteristics of the organism are monogenously controlled. Uh, it looks like one pair of allelic genes. Non-allelic genes are genes located in different regions of chromosomes and encoding different proteins. Non-allelic genes can interact with each other in all cases of gene interactions. Mendelian patterns are strictly observed, with either of gene causing the development of several traits, or conversely, one trait manifests itself under the action of a combination of several genes. The interaction of non-allelic genes is manifested in four main forms: epistasis, complementarity, polymerization, and pleiotropy. Genotype is a set of genes of a given organism. The genotype, in contrast to the concept of the gene pool, characterizes an individual, not a species. In a narrower sense, a genotype is understood as a combination of alleles of a gene or locus in a particular organism. This process is called genotyping. The genotype together with environmental factors, determines the phenotype of the organism. In this case, individuals with different genotypes may have the same phenotype, and individuals with the same genotype may differ from each other in different conditions. Numerous experiments have confirmed the correctness of the regularities established by Mendel. At the same time, facts appeared showing that the numerical ratios obtained by Mendel when splitting the hybrid generation were not always observed. This indicated that the relationship between genes and traits was more complex. It turned out that one and the same gene can influence the development of several traits. One and the same trait can develop under the influence of many genes. It should be noted that the interaction of genes is of a biochemical nature. That is, it is not genes that interact with each other, but their products. The product of an eukaryotic gene can be either a polypeptide or transport RNA or ribosomic RNA. Unless different forms of the same gene located in the same areas of homologous chromosomes determine the direction of development of a particular trait. In a diploid organism, there can be two identical alleles of the same gene. In this case, the organism is called homozygous, or two different ones, which leads to a heterozygous organism. Normal diploid somatic cells contain two alleles of one gene according to the number of homologous chromosomes, and haploid gametes contain only one allele of each gene. For traits that obey Mendel's laws, dominant and recessive alleles can be considered. If the genotype of an individual contains two different alleles, an individual is a heterozygote, the manifestation of a trait depends only on one of them, the dominant one. The recessive allele affects the phenotype only if it is located in both chromosomes. The individual is homozygote. Thus, the dominant allele suppresses the recessive one. In more complex cases, other types of allelic interaction are observed. However, it should be noted that despite the variety of interactions of alleles, sometimes very complex, they all obey Mendel's first law, the law of uniformity of first-generation hybrids. Soon after the rediscovery of Mendel's laws, it was established that the interaction of genes is not limited to the complete dominance of the dominant allele over the recessive one. In fact, there are complex or varied interactions between the alleles of one gene and different genes in the genotype, which play an important role in the implementation of the information contained in each individual gene. Dominance means that one of the alleles of the pair masks of completely suppresses the expression of the second allele, recessive. At the same time, the degree of suppression of the recessive trait determines various variants of dominance. Kinds of interaction of allele genes distinguish between complete dominance, incomplete dominance, codominance, allelic exclusion. Complete dominance. is a type of interaction of allelic genes in which the phenotype of heterozygotes does not differ from the phenotype of homozygotes for the dominant. That is, the product of the dominant gene is present in the phenotype of heterozygotes. Complete domination is widespread in nature. Takes place when inheriting, for example, the color of shape of pea seeds, eye color, and hair color in humans, RH antigen, and many others. The presence of the RH antigen, RH rhesus factor, of erythrocytes is due to the dominant RH gene. That is the genotype of a RH positive person uh, can be of two types. The first, either RH, RH, or uh, RH, R small, letter small, H. Uh, if, for example, the mother is RH negative and the father is RH positive and heterozygous, for this trait, then with this type of marriage, both RH positive and RH negative children can be born with the same probability. Probability. An RH conflict, rhesus conflict, can arise between a rhesus positive fetus and an rhesus negative mother. Incomplete dominance. This is the name of the type of interaction of allelic genes in which the phenotype of heterozygotes differs both from the phenotype of homozygotes for the dominant and from the phenotype of homozygotes for the recessive and has an average intermediate value between them. Occurs when the color of the appearance of a night beauty, uh, snapdragon hair color of guinea pigs, etc., is inherited. Mendel himself faced incomplete dominance when he crossed a large-leaf pea variety with a small-leafed one. The hybrids of the first generation didn't repeat the trait of any of their parent plants. They had medium-sized leaves. When homozygous red fruit and white fruit strawberry varieties are crossed, all the first generation of hybrids have a pink fruits. The correspondence of cleavage by genotype to cleavage by phenotype is characteristic for incomplete dominance, seen heterozygotes and phenotypically different from homozygotes. Codominating. Codominating is a type of interaction of allelic genes in which the phenotype of heterozygotes differs both from the phenotype of homozygotes for the dominant and from the phenotype of homozygotes for recessive. And the phenotype of heterozygotes contains the products of both genes. Occurs during the formation, for example, of the four group blood of the system AB0 in humans. In order to imagine how the inheritance of blood groups in humans occurs, you can see the birth of children with which blood group is possible in parents who have one, the second, uh, the other, the third blood group, and who are heterozygous for this trait. Overdominance. Overdominance is a phenomenon of the advantage of a class of heterozygotes in comparison with the classes of homozygotes possible for a given gene and alleles. Phenotypically, as a rule, in the case of overdominance, heterozygotes don't have special external characteristics. The advantage is related to biochemical characteristics. One of the characteristic examples of overdominance is the increased frequency of the allele of the sickle cell anemia gene in human populations living with a high probability of contracting malaria. The mutant allele protects the body from malaria. Homozygotes for the normal allele can contract malaria and die. Homozygotes from the mutant allele are more likely to die from anemia. Heterozygotes for this gene do not suffer from sickle cell anemia and are resistant to malaria. Like any phenomenon that leads to a change in the fitness of individuals in populations, overdominance is associated with a genetic burden. More adapted heterozygous organisms, when crossed both with each other and with representatives of other genetic classes, should give less adapted offspring. The genetic burden associated with maintaining genetic diversity is in a population under overdominance is called segregation. An extreme case of overdominance is a complete non-viability of homozygotes. The consequence of the model is that in natural population, overdominance cannot uh, simultaneously give great advantages to heterozygotes and spread to a large number of genes. Otherwise, the payment for the increases fitness of some individuals will be the need to maintain fertility at an unattainable level. Inter-allelic complementation. Inter-allelic complementation is a rather rare interaction of alleles. It occurs when a normal G trait can be formed in an organism heterozygous for two mutant D1, D2 alleles. It has been found that all genes whose uh, alleles interact in this way control the structure of enzymes built from the same protein subunits. If the mutant alleles differ in that they encode polypeptides of different domains, then when they are combined in a heterozygote, all, all, all the necessary functional centers will be collected in the enzyme molecule, since mutant alleles are responsible for the defectiveness of only some of them. Then the enzymatic activity, which means and the normal phenotype will be restored. Penetrance. Penetrance is an indicator of the phenotypic manifestation of an allele in a population. It is defined as the ratio of the number of individuals in whom phenotypic manifestations of the presence of an allele are observed, to the total number of individuals in which this allele is present in the number of copies necessary for phenotypic manifestation. For example, the phrase allele A has a penetrance of 95% means that of all individuals in which this allele is present in the required number of copies, only 95% of the presence of this allele can be determined by phenotype indicators. Full penetrance in 100% phenotypic manifestation of the presence of a given allele within a population. In simpler terms, it is frequency of a gene's expression in traits. The term penetrance was proposed in the 1926 by Timofey Frysovsky and Vogt. In medicine, penetrance is the proportion of people with a given genotype who have at least one symptom of a disease. In other words, penetrance determines the uh, likelihood of a disease, but not its severity. Some believe that penetrance changes with age, such as in Huntington disease, but difference in age at onset are usually attributed to variable expressively. Sometimes penetrance depends on environmental factors. Penetrance can be important in medical genetic counseling for autosomal dominant diseases. A healthy person with one of the parents suffering from a similar disease, from the point of view of classical inheritance, cannot be a carrier of a mutant gene. However, if we take into account the possibility of incomplete penetrance, then the picture of completely different. An outwardly healthy person can have a non-manifest mutant gene and pass it to children. Gene diagnostic methods can determine whether a person has a mutant gene and distinguish a normal gene from a non-manifest mutant gene. Expressiveness is a degree of phenotypic manifestation of an allele. If the variability of the trait for which the allele is responsible is absent, the expressiveness of such a trait is constant. Well expressed variability variability of the trait indicates a changeable of variable expressivity. To assess the manifestation of a trait controlled by alleles with high expressiveness. An additional trait is used penetrance. For example, alleles of AB zero blood groups in humans have constant expressivity, always 100, and alleles that determine a color have variable expressivity. Penetrance is a qualitative phenomenon. Uh, Expressiveness is quantitative. Expressiveness reflects the nature and severity of symptoms as well as the age of onset. Patients from the same family with one mutation may have hyperplasia or neoplasia of one of all endocrine tissues, including the pancreas, parathyroid glands, pituitary gland, and adipose tissue. As a result, the clinical picture of the disease is extremely diverse. In patients from the same family, peptic ulcer disease, hyperglycemia, urolithiasis, or pituitary tumors can be found. Sometimes in dominant diseases characterized by the formation of tumors, differences in expressivity are cases by additional mutation in tumor suppressor genes. If a person suffering from a dominant disease wants to know how difficult the disease will be in his child who inherited the mutation, then he raises the question of expressiveness. With the help of gene diagnostics, it is possible to identify a mutation that does not even manifest itself. But it is impossible to predict the range of expressiveness of the mutation in a given family. With the pleiotropic action of the gene, one gene determines of the development or affects the manifestation of several symptoms. This property of genes has been well studied in the example of dwarfism in mice. It was shown that when phenotypically normal mice were crossed in F1, mice were dwarf offspring, from which it was concluded that dwarfism is due to a recessive gene. Recessive action genes modifiers homozygotes stopped growing in the second week were enabled to reproduction the internal organs, especially the endocrine glands, had an altered shape. The mice were less mobile and poorly tolerated temperature extremes. The gene for dwarfism determined the abnormal development of the pituitary gland, which in turn determined early growth arrest. Abnormal development of the thyroid gland, which determined a reduced metabolism. Therefore, dwarf mice were sensitive to cold but more resistant to hunger. This is a chain of sequential changes in traits with a defect in only one gene. An example of the pleiotropic action of a gene in humans is inheritance defect of nails and defect of the patella, for which is responsible of gene. An example of a house sublettal gene is a gene that determines a disease retinoblastoma, in which cancer develops in early childhood swelling of the eye. Previously, this disease had always led to death, and now they carry out operations that save a person but lead to blindness in one or both eyes. Uh, in the action of the recessive gene of phenylketonuria leads to an increases content of phenylalanine in the blood. It decreases in mental development, change in head size and hair color. Action of the dominant gene spider fingers, uh, arachnodactyly or Marfan syndrome. At the same time, heart disease and lens abnormalities appear as eyes. In Pakistan, some people do not have sweat glands on certain parts of the body. This correlates with missing teeth. The development of a trait is determined by two alleles of one gene, a letter A big and letter A small, which occupy identical loci of homologous chromosomes. Sometimes a gene has not two, but more alleles that arise as a result of mutation. Multiple mutations of the same gene form a series multiple alleles, and the phenomenon itself is called the phenomenon of multiple allelism. It is widespread coat color in rabbits, eye color in Drosophila, AB0 blood group system in humans. There are certain patterns of multiple alleles. Each gene can be a large number of alleles. Any allele can result from direct and reverse mutation of any member of a series of multiple alleles or from a wild-type allele. In a diploid organism, any two alleles from a series of multiple alleles can be uh, sim- simultaneously present. Alleles are in complex dominant recessive relationship with each other. One and the same allele can be dominant with respect to one allele and recessive with respect to another. And between other alleles, dominance can be absent, and codominance is observed. Members of a series multiple alleles are inherited in the same way as a pair of alleles, that is, inheritance obeys Mendel laws, except for codominance. Different combination of alleles in the genotype causes different phenotypic manifestations of the same trait. A series of alleles increase the plant's necessary variability. An example of multiple allelism in humans, it is the presence of three alleles of a gene that determines the inheritance of blood group in the AB0 system. The system is determined by three alleles of one gene. 1. Gene 1 is located on chromosome 9, 9q34. From the entry series of alleles, two alleles are simultaneously present in the genotype of a diploid organism. The dominant allele of the gene can expect it is effect of homozygous and heterozygous organism, and the recessive allele of the gene only in a homozygous organism. Different combination of alleles in the genotype give different phenotypes for blood groups 1, 2, 3, 4, which differ in the antigenic properties of erythrocytes. Antigens, agglutinogens, are located on the surface of erythrocytes, glycocalyx. A feature of the system is the presence in the blood serum of specific antibodies, agglutinins, which are unlike the oven agglutinogens, they simultaneously in the blood. Blood groups are an example of ambiguous, an ambiguous norm of the body's reaction. The blood group does not change during life under the changes in the environment. In Europe, 85% of people have an antigen protein of the surface of erythrocytes, which is called the rhesus factor, a rash factor. They have conventionally called RH positive. 15% have no such antigen in the erythrocytes. They conventionally called RH negative. The presence of an antigen is determined by their closely linked genes. C, D, K, chromosome 1, P35. They are inherited together as one gene and are designed by one symbol RH. The RH factor is inherited according to dominant type. Sometimes with blood transfusion and some types of marriages, a rush incom- incompatibility occurs. A classic example of incompatibility for the RH factor of the mother and the fetus is an option when the RH positive fetus develops in the body of RH negative mother. Uh, for the prevention of hemolytic diseases in subsequent children, women is injected with anti-rash antibodies before of immediately after childbirth, uh, which blind antigens fetal erythrocytes that have entered the bloodstream. Clark affects prevention of rash incompatibility. The mother immune system remains intact. Determination of blood groups of the AB0 and rhesus system is used in the study of twins. Determination is concordance according to the studied the trait in pairs of monozygotic and dizygotic twins. Forensic medical examination, blood transfusion, transplantation, paternity. To study blood group system, you need know immunogenetics, the structure of antigens, the relationship between antigens and antibodies, the possibility of agglutination of erythrocytes in case of incompatibility by blood groups of the donor and recipient, pregnant woman and fetus. Non-allelic genes are genes located in different regions of chromosomes uh, and encoding different proteins. Non-allelic genes can also interact with each other. In this case, either of one gene determines the development of several traits, or conversely, one trait manifests itself under the influence of a set of several genes. There are three forms and interaction of non-allelic genes. Complementary action genes. Genes are called complementary when in their manifestation they seem completed each other. Each such gene, individually by itself, does not have an effect, does not cause the appearance of a trait. However, when two individuals are crossed, each of which has this type of a gene. For example, one has gene A and the other has gene B. A hybrid obtained a new organism in which both of these genes are available, and the effect of their joint action is manifested. It's that under the joint influence, a new sign appears in a hybrid organism. Epistatic action of gene epistasis. The epistatic action of genes is by its nature oppositive to the complementarity action of genes. Its essence lies in the suppression of the action of hypostatic genes by suppressor genes. The phenomenon of epistasis is expressed in non-allelic suppression of the action of one dominant gene by another dominant gene belonging to another allelomorphic pair. Genes that suppress the action of other non-allelic genes belonging to other pairs are called epistatic. Epistasis is expressed in a change in the ratio of cleavage in the second generation, which phenotypically deviates from the usual cleavage in dihybrid crossing, that is from Mendelian norm 9 to 3 to 3 1. Currently, epistasis is divided in two types, dominant and recessive. Dominant epistasis is understood as suppression by the dominant allele of one gene of the action of the allelic pair or another gene. Uh, an interesting example of reticent epistasis is Bombay blood phenotype. In rare cases resident of Bombay, India, in genotype of which the dominant alleles 1A or 1B group blood is 0. It was found that this defect is caused by recessive mutation in H gene, not homologous to loci A or B, which in a homozygous state leads to Z in the process of synthesis of A or B antigens as a defective H substance with which the glyco glycosyl transferase enzyme cannot interact and therefore normal antigens are not formed. In this case, in a digibrid crossing, the phenotype splitting will correspond to 9 to 7, as in the case complementary gene interaction. Therefore, one and the same attitude can be interpreted as a complementary interaction and as epistation. Genetic analysis itself inheritance in the interaction of gene without taking into account of biochemistry and physiologically of the development of a trait in ontogenesis cannot reveal the nature of interaction, but without genetic analysis, it is impossible to understand the hereditary determination of the development of these traits. Polymerism is a phenomenon of gene interaction in which several genes uh, of the same type have a similar effect on the development of the same trait. In other words, polymerization is caused by the action of a different number of unambiguous genes which, when summed up, enhance the manifestation of a trait and with a smaller number of such genes. This trait is manifested to a correspondingly lesser extent. The study of the inheritance of quantitatively varying traits in divergent individuals of the same generation began in the first decade of the 20th century. In medical genetics, if all carriers of a pathological gene have its clinical phenotypic manifestation, that we can talk about 100% penetrance. If the effect of the mutant gene is manifested not in all of its carriers, incomplete penetrance. In this case, the carrier of the pathological gene may be clinically healthy, and there is a generation skipping in the pedigree. An indicator of the dependence of an individual gene on the entire genotype is the expressivity of a gene, the severity of one and the same trait in organisms that have a gene that controls this trait. Talking about the expressiveness of a gene that causes hereditary diseases. Talking about the severity of the disease. Thanks for your attention, students. The literature take and the slide.